Hello, welcome back to the A&P Madhouse with Dr. H. Today we're going to be covering even more cardiovascular physiology. Today we're going to be covering the baroreceptor reflex, however. And what the baroreceptor reflex is all about is homeostasis, homeostasis, homeostasis. Hopefully you guys are starting to catch that in A&P, right? Anatomy and physiology. The human works via homeostasis. So if blood pressure gets too high, what's the body going to try to do to fix it? It's going to try to bring it back down. And if blood pressure gets too low, what's the body going to try to do? It's going to try to fix it. It's going to try to bring it back up. And it's going to have some abilities to be able to fix blood pressure right now. Within 200 to 500 milliseconds, we're going to start getting changes to blood pressure. How does the body do this? Well, it uses baroreceptor reflexes. All right. What a baroreceptor reflex is, what the baroreceptors themselves are, baro is pressure receptors. Okay, we have pressure receptors in multiple locations, but some of the best places for measuring a blood pressure is right here in the aortic arch. So we're going to have a subset of baroreceptor receptors, I guess I should say, baroreceptors right here in the carotid. I just said carotid, right here in the aortic arch. Okay, so the first location is in a portion of the aortic arch called the aortic sinus. We're just going to go ahead and call it the aortic arch. The second location that we find these baroreceptors is way up here in the sinus. Okay, so we're going to actually find this up here in the carotid sinus as well. So if you started to go through blood vessels already, you'll know that the aortic arch is going to branch into the brachiocephalic trunk, which splits into the common carotid artery. This is the right common carotid artery, and the common is going to split again into an internal and external carotid. And that is where we find this sinus. On the left-hand side, we don't have this fused portion of the brachiocephalic trunk. Rather, coming off the aortic arch, we simply have the left common carotid artery. And up here at the branch between the internal and external carotid arteries, well, we're going to have these baroreceptors. And the way these baroreceptors work is they are going to be triggered by the stretch itself. The baroreceptor is a mechanoreceptor. Mechanoreceptors means that they are changed by physical properties. So what's going to happen is we basically are just going to have, we're just drawing an artificial cell here, a nice simple artificial cell. And if this cell gets stretched, what this cell has in the cell membrane is a sodium channel that will be opened up by that stretch. And so if we have a sodium, a mechanically gated channel, right? Mechanically gated, but it's a mechanically gated sodium channel, then what do we know? As this gate gets open, sodium, which is higher concentration outside of the cell, is going to now leak into that cell. And what's going to happen to the membrane potential inside of the cell? It's going to increase. And as we depolarize the inside of these cells, it's going to end up sending an action potential to where it needs to go. So if we're looking at the baroreceptors down here in the aortic arch, what we're going to see is that these are going to, in essence, send off an axon. Let's do it in a different color so we don't get this all mixed up. Right? These are going to send off an axon that are going to go all the way up to the brain. Okay, they're going to go all the way up to the brain. And they're going to go all the way to the medulla oblongata, where they have their synapses. Let's just add those together. All right, this right here that they're communicating to the brain through is the vagus nerve, probably one of the most important nerves in relation to the autonomic nervous system and to the parasympathetic nervous system. We'll see how that works out. All right, so this is vagus nerve, or the cranial nerve number 10. Okay, that's the vagus nerve right there. Well, what about up here? What about up here in this carotid sinus? What nerve is communicating to the brain here? Well, this is actually going to be, and we'll go ahead and bring these all together. This nerve right here is going to go up here as well to the same location in the medulla oblongata. But this is the glossopharyngeal nerve. So this is cranial nerve 9. Okay, that's the glossopharyngeal nerve that is going to send those carotid sinus action potentials to the medulla oblongata. And up here in the medulla oblongata, there are 3 little subnuclei. Okay, and we're going to go ahead and kind of draw each one in just as an independent. This is not the exact location that we find these, just trying to separate them out for you. Okay, 1, 2, we'll do the last one in here in yellow, 3. All right, so if we want to try to list these things out, well, let's first group them all together. Okay, these 3 subnuclei of the medulla oblongata combined are called the cardiovascular control center. The cardiovascular control center, which we can abbreviate the CVCC, the cardiovascular control center. And the cardiovascular control center is going to have 3 subnuclei. 1, we're going to have a cardioacceleratory. All right, acceleration means to speed up. So it's going to have a cardioacceleratory center. 2, it's going to have a cardioinhibitory center. Which is going to slow that heart down. And 3, well, it's going to have some vascular control, so control over blood vessels. We call this vasovascular motor. Control of the muscles, so a vasomotor center. Okay, cardioacceleratory, cardioinhibitory, and the vasomotor. So let's go ahead and label those. We'll go ahead and call this first one up here the cardioacceleratory. So we'll say this one is CA in purple. The next one down, we'll go ahead and call our CI, cardioinhibitory. And then this one down here will be the vasomotor. So we're going to go ahead and just call it VM. Now, let's go ahead and continue this idea. This information from the cardioacceleratory and cardioinhibitory and the vasomotor centers, well, they're going to go back out to the body. Okay, cardioacceleratory is going to try to improve heart function. Okay, what part of our sympathetic nervous system improves heart function? And hopefully you guys can see. Let me use a different color yet. All right, we'll go ahead and do a nice dark green here. Okay, that this now is going to be acting through the sympathetic nervous system. And the sympathetic nervous system has a relatively short preganglionic neuron, and then its synapses, and then it's got a much longer postganglionic neuron. And that neuron down here is going to end up synapsing in where? Okay, where do we find receptors for the sympathetic nervous system on the heart? Well, we find them on the SA node. I'm kind of running out of colors here, but that's okay. We find them on the SA node. We find beta 1 adrenergic receptors up here. Where else do we find receptors? We find them on the AV node. Beta 1 adrenergic receptors there as well. And then we find them all over the myocardium. They're all over down there, right? So if we follow out, where is this going to go and communicate with? Well, it's going to go and communicate with the SA node, the AV node, and then it's also going to send information out all over the place. Okay, all over the myocardium. This is important in terms of the fact that now we know that the sympathetic nervous system can change the autorhythmic cells and it can change the myocardial cells. So let's look at what those can do, right? So if we're talking about the autorhythmic cells, if you've seen my previous videos, you should already know this. We know that the autorhythmic cells control the heart rate versus the myocardial cells, which now control the strength at that heart beats, and that's controlling the stroke volume. Cool. So the sympathetic nervous system can increase the heart rate and it can change the strength of the stroke volume. How the heck does it do that? We're going to come back to that in just a second. What else can the sympathetic nervous system do? The sympathetic nervous system also communicates with blood vessels. So let's go ahead and have a second neuron over here in our sympathetic chain ganglia. And we're going to have this go down and it's going to communicate with, that's not with the direction. All right. We're going to have it come down here and it's going to communicate with blood vessels. So let's draw out one blood vessel and we'll go ahead and have this one a little bit smaller than the other one. And we'll have some arrows to depict that we're going to end up causing vasoconstriction in this particular blood vessel. Whereas the other blood vessel down here, we can potentially, and we're not going to have direct innervation of this blood vessel, but the sympathetic nervous system is going to act on it, and it's going to end up with, that's supposed to be an arrow, vasodilation. All right, so these are going to be blood vessels. Hopefully you guys remember that most of the blood vessels of the human body are covered in alpha-1 adrenergic receptors. Alpha-1 adrenergic. And what happens with alpha-1 adrenergic receptors? When the sympathetic nervous system is activated, now we're going to be releasing norepinephrine, and that's going to land on most blood vessels of the human body. The norepinephrine is going to activate alpha-1 adrenergic receptors. Pretty cool. Alpha-1 adrenergic receptors are excitatory, which is going to end up resulting in vasoconstriction. So what just happened to the radius of this tube? The radius got smaller, and when we vasoconstrict, what's going to end up happening is we're going to affect resistance. So as we vasoconstrict, this is going to end up increasing the resistance, but not just any resistance. We're talking about the resistance throughout the entire body. This is total peripheral resistance. So the TPR, the total peripheral resistance, starts to increase as the sympathetic nervous system communicates with all of these blood vessels. In addition to alpha-1 adrenergic receptors on blood vessels, we also have beta-2 adrenergic. But here's the deal with beta-2 adrenergic receptors, is beta-2 adrenergic receptors are inhibitory. So when beta-2 adrenergic receptors activate, it is going to inhibit the muscles of the blood vessels. So it's going to cause those blood vessels to vasodilate. And that vasodilation is going to increase the radius, decreasing the overall resistance. Pretty interesting stuff. Now, beta-2 adrenergic receptors and alpha-1 adrenergic receptors, where do we find them? Remember, alpha-1, I already said it, are found most of the locations in the human body. Beta-2, unfortunately, are only found, well, fortunately, they're only found on the blood vessels covering the heart, the coronary arteries, and they're found in the blood vessels of the skeletal muscles. So if we end up with sympathetic stimulation, it's going to shut off blood supply to most of the organs of the human body, which is great because in a sympathetic response, we want more blood going to our heart so it can beat more efficiently and our skeletal muscles so we can run away from Mr. Axeman. That's why we have alpha-1 and beta-2. It allows those different functions of the body to work appropriately. Cool. Now, in relationship to this sympathetic stimulation of the heart, let's go ahead and go down here and we're going to summarize that real quick, right? So if we end up with sympathetic stimulation, so sympathetic nervous system on autorhythmic cells, so we're going to go ahead and say autorhythmic cells, so auto, let me write that in, cells. Then what we're dealing with is the sympathetic nervous system. What do we remember about it? It's releasing norepinephrine, potentially epinephrine if the medulla, if the adrenal medulla has been activated. Now those are going to land on those autorhythmic cells. And how do they act? Well, they're going to activate beta-1 adrenergic receptors. What happens with those beta-1 adrenergic receptors? Well, they are GPCRs. So we're going to get an activation of a G stimulatory protein. That's going to go and act on adenylate cyclase. And as we activate adenylate cyclase, that's going to convert ATP. It's going to hydrolyze ATP into cyclic AMP. And then that cyclic AMP is going to go and increase the activity of pKa, and pKa is going to go and open up funny channels. So that increases the sodium influx. And it's also going to act on the T-type and the L-type calcium channels. So now we increase calcium influx. All of which does what? All of which increases the speed that depolarization of autorhythmic cells have. So it increases the rate of depolarization. And then what? What happens to the heart rate? We'll go ahead and come over here. That's going to increase the heart rate. So we know the sympathetic activity on heart rate is to increase the activity of that heart rate. What about the sympathetic activity of the myocardial cells? Again, I'm just going to try to summarize all this for you guys so you can see how this all works together. So now if we're dealing with myocardium or myocardial cells, it's going to be doing a very similar thing where you still have norepinephrine and epinephrine. You still have beta-1 adrenergic receptors. You still are activating this G stimulatory protein, which talks to adenylate cyclase and goes on to increase cyclic AMP and pKa. So all of that still occurs. So we're going to go ahead and say same as autorhythmic, right? So let's just start here at an increase in cyclic AMP. We know that in myocardial cells, we're still going to get an increase in cyclic AMP, and that is going to increase pKa. And pKa goes around and phosphorylates things. So what is pKa going to phosphorylate? pKa is going to phosphorylate the L-type calcium channels. So we're going to throw a little phosphate group on those L-type calcium channels. And PK is going to go and phosphorylate the calcium pumps found on the sarcoplasmic reticulum and on the cell membrane. So now we're going to phosphorylate those calcium pumps. That's going to open the L-type calcium channels a little bit longer, which ends up resulting in an increase in the concentration of calcium inside of that cell. And what does that do? That increases the amount of calcium binding to troponin, allowing for an increase in the force of contraction. And then the calcium pumps, well, now we're going to have a greater concentration of calcium that's going to move into the ICF because we're pumping that calcium in. Don't forget, ryanodine receptors are open this whole time. So now that calcium goes out into the ICF. And if we turn that ryanodine receptor off, then the calcium is removed from the ICF faster, and that's going to increase the rate of relaxation as well. So increases the rate of relaxation, and it increases the force of contraction. What determines which pathway this goes on is whether or not the ryanodine receptors are open. So if the RYR are closed, well, then that calcium just gets pumped out of the ICF faster. But if the RYR is open, then that calcium just gets to move out of that sarcoplasmic reticulum faster, and that's going to join that calcium with the rest of the calcium in the ICF, binding to troponin, doing what it do, right? Increasing the force, the strength of contraction. It's kind of nice being able to throw all this together on one picture. I hope you guys are following along because a lot of this should be review. How would the parasympathetic nervous system act on this entire thing? All right, let's go back up here. And actually, I didn't do something quite right, and I apologize. Because what do we know about the sympathetic nervous system that's talking to the blood vessels? That is coming from the vasomotor center. So let's go ahead and have that go up to that vasomotor center. It's the vasomotor that is affecting our blood vessels. I apologize for not saying that earlier. I made the assumption that you guys followed me, but I realized that I should always have it in the diagram. So there you go. The last part of this diagram that we're going to go ahead and follow using the color dark blue is from the cardioinhibitory. Okay, cardioinhibitory now is the parasympathetic nervous system. Rest and digest is what slows that heart down. So now this is going to go and act where? Where do we find receptors for the parasympathetic nervous system? Only on the autorhythmic cells, and that is it. Okay, there are no muscarinic receptors on myocardium. So the parasympathetic nervous system, go ahead and do this green so you know that I'm talking about the SNS here. The parasympathetic nervous system acts down here, and I didn't draw something in quite right because the parasympathetic nervous system has a long preganglionic neuron that's going to synapse down here on a short postganglionic neuron. And then that parasympathetic nervous system is going to be secreting. Nice thing about most of this is sympathetic, right? Sympathetic nervous system, sympathetic nervous system, sympathetic nervous system. Parasympathetic nervous system only affects autorhythmic cells. That's it. So when we try to look at what the parasympathetic nervous system is going to do, well, we're going to end up saying what neurotransmitter is released from the parasympathetic nervous system? Acetylcholine. What does acetylcholine act on? Muscarinic receptors. What do muscarinic receptors do? Well, they are GPCRs, and those GPCRs are going to have basically an excitatory portion of them and an inhibitory portion of them. There's an alpha, a beta, and a gamma. The beta and the gamma work together, and the alpha does what alphas do. So the beta and the gamma, well, they're going to go and open up and this is called beta inhibitory. So muscarinic receptors, if we're talking about G proteins, we're going to have a beta inhibitory and a gamma inhibitory. They go and they hyperpolarize the cell. How do they hyperpolarize the cell? They open up a T-type or a transient potassium channel. Super cool, super interesting, this little transient guy right here. It's completely going to change how this cell membrane responds because it's going to cause hyperpolarization of that cell membrane. And when we hyperpolarize that cell membrane, we make it go further from threshold. What else is going to happen is that this G protein, there's an alpha subunit. It's an alpha inhibitory subunit. It's supposed to be an I, sorry. Alpha inhibitory subunit. This is going to go and it's going to actually block pKa's function. So this is going to decrease pKa function. And as you decrease pKa function, you're going to close those funny channels, those T-type calcium channels, and that is going to decrease sodium influx. It's going to decrease calcium influx, all of which is going to decrease the rate of depolarization, which is what we're after, because ultimately what does that do? The combination of hyperpolarization and decreasing the rate of depolarization, those two things together, what is that going to do? It's going to decrease the heart rate. Awesome. Let's pull this all together now, because that's what this has all been about. Let's pull this all together, and I'm going to use this tiny little space right here. This is going to be my insert that I get to use. Not a lot of space to put it all together, but that's how the human body works, right? We tend to need a lot of background information to be able to get to the text we are after. Because ultimately what we're after is controlling blood pressure. That's what I said baroreceptors do to begin with. So we want to control that blood pressure. And blood pressure is mean arterial pressure. MAP, M-A-P, mean arterial pressure. And we know that MAP is proportional to, I might have to go back to a previous video of mine, cardiac output times TPR. Those are going to be two of the most important variables that we're going to be dealing with. In addition, one other variable that I personally like to discuss is blood volume. Blood volume could potentially have an impact on cardiac output, but we're going to still separate it from the rest of this cardiac output function. So this is what we've been after this whole time, is what is the body going to try to do if the mean arterial blood pressure decreases or if it increases? All right, so what do we know? If mean arterial blood pressure decreases, patient has hypovolemia, has diabetes, and is peeing out glucose and has polyuria because water follows that glucose. Maybe the patient is dehydrated. Maybe the patient's taking a diuretic. Who knows? Whatever the case may be, their mean arterial blood pressure is going to be decreasing. What do we know the body's going to try to do in this particular example? We know that in one example, if we have a decrease in mean arterial blood pressure, that's the stimulus. So we're going to say there's a stimulus of a decrease in mean arterial blood pressure. This is hypotension. Or we can have a stimulus, which is an increase in mean arterial blood pressure, which would be basically hypertension, right? What are some immediate responses the body can do to try to fix this problem? The baroreceptor is going to kick in, and the response is going to be opposite of the stimulus. So what's the body going to try to do? Increase that mean arterial blood pressure. What's the body going to try to do if we become hypertensive? It's going to try to decrease mean arterial blood pressure. Remember, if it can't fix this, that's what a pathophysiology is all about. That's a disease. So how can the body do this? Watch. It's pretty simple now. Blood pressure drops. Less blood makes it to the aortic arch. Less blood is pushing against the walls of the carotid sinus. What's going to end up happening is because blood pressure dropped, these mechanoreceptors stop stretching and they stop depolarizing. Less sodium enters, they slow down their rate of depolarization. That's going to decrease the number of action potentials being sent to the medulla oblongata via the vagus nerve and the glossopharyngeal nerve. And as the stimulus decreases to the medulla oblongata, what does the body want to try to do? As mean arterial blood pressure dropped, we know it's going to try to pick things up again. So as less action potentials go to the medulla oblongata, what's going to happen to the sympathetic nervous system? We're going to activate that sympathetic nervous system. And as we activate that sympathetic nervous system, what's going to happen? One, as we activate that sympathetic nervous system, heart rate is going to increase. Two, as we activate that sympathetic nervous system, myocardial cells are going to increase their force of contraction and their rate of relaxation so they can pump stronger, faster, and relax faster. Three, we're going to go down to these blood vessels, and the alpha-1 adrenergic receptors are going to vasoconstrict, thus increasing TPR. Wow. So heart rate, stroke volume, and I actually didn't finish that off over here. Let's write that in just so it's there. Force of contraction is related to stroke volume. So we'll go ahead and underline that in green too. And what do we know about cardiac output? Cardiac output equals heart rate times stroke volume. So as the heart rate goes up, the stroke volume goes up, the cardiac output will go up. And as the sympathetic activity of the alpha-1 adrenergic receptors increases, then it vasoconstricts and increases the resistance. The response is to try to bring that mean arterial blood pressure back up, and that's exactly what we just saw. As a compensatory response, cardiac output will increase, TPR will increase to try to fix the problem. What about if mean arterial blood pressure gets too high? Right? If mean arterial blood pressure gets too high, this will work. If mean arterial blood pressure gets too high, then the amount of blood pressure in the aortic arch and in the carotid sinus increases, which now stretches the mechanoreceptor so that sodium leaks in faster, depolarizing the baroreceptors faster, sending more action potentials through the vagus nerve, through the glossopharyngeal nerve, to the CVCC. Within the CVCC, what do we want to do? Well, we want to decrease the mean arterial blood pressure. So which of these are we going to activate? We're going to activate the parasympathetic nervous system, aren't we? What else are we going to have to do, though? we can't just activate the parasympathetic nervous system. We also need to inhibit the sympathetic nervous system. And so the increase in action potentials to the CBCC also inhibits the sympathetic nervous system, and its signals to the heart rate is going to inhibit the sympathetic nervous system to the myocardial cells, and it's going to inhibit the sympathetic innervation of the blood vessels. And if we inhibit vasoconstriction, what's going to happen to this blood vessel? It will vasodilate. And as it vasodilates, what happens to TPR? TPR will decrease. So what happened to cardiac output? That heart rate slowed down. We got rid of sympathetic activity, so the stroke volume did decrease, but not due to a direct effect of the parasympathetic nervous system. And the TPR is going to decrease as well, thus bringing that map back down to normal. All right, hopefully I was able to summarize the baroreceptor reflex for anybody that was confused by it, gave you a lot of that background in review so that we can tie this all together. Please, please, please watch it again if you need to, rewind it to where you need to go to in order to understand this. Reach out, ask questions below. All right, throw down a like if you liked it. In my next video, I will talk about something else related to heart, and that's going to be probably the intrinsic and extrinsic controls of mean arterial blood pressure, because the one part of this equation we haven't talked about yet is how does the body go about controlling blood volume. All right, this is the end of the madness with Dr. H. Humankind be both.